Hello everybody and welcome to this latest episode of Cardiomyopathy Conversations. It's Libby here and I'm delighted today to be joined by Professor Ruth Newbury-Ecob, clinical geneticist, and Professor Perry Elliott, consultant cardiologist. Today we're going to be talking all about genetics and cardiomyopathy. So guys, welcome to the podcast.
Nice to be here.
Do you like my new surroundings?
Love it.
Nice new gaff. We've gone up in the world. So I thought we'd start by doing some quick fire questions. Okay. Now, I'm going to start with you, Ruth.
Okay.
And it's a yes or no answer. Okay. You're allowed to say it depends, but only once. Okay. Are you ready?
I'm ready.
Okay. Are all cardiomyopathies genetic?
No.
Is genetic testing useful in identifying at-risk family members?
Yes.
Can someone carry a cardiomyopathy gene without developing the disease?
Yes.
Do lifestyle factors influence the expression of genetic cardiomyopathies?
Yes.
Sure?
Yes.
Yes. Okay. Is hypertrophic cardiomyopathy the most common inherited heart condition?
No.
Can a negative genetic test completely rule out inherited cardiomyopathy?
Am I allowed to say tricky?
Well, you're allowed to say it depends, but only once.
I'll take my depends for that
Okay,
one.
depends. Do all gene variants found on testing have clear clinical meaning?
No.
No. Can cardiomyopathy skip generations? No more it depends, Ruth.
Can I say it depends?
Yeah.
You can, but you forfeit your it depends for your bit.
Okay. Do I come to your rescue, Ruth?
the question, it's define skip.
Okay. Well done. Perry. True or false? Okay. It depends only once.
Okay.
If you have the gene for a cardiomyopathy, you're guaranteed to develop the disease.
False.
A normal echo means you don't need genetic testing.
False.
Genetic counselling is only important after testing.
False.
A variant of uncertain significance always means disease.
False.
Only athletes are at risk of sudden cardiac death from cardiomyopathy.
False.
I should have put some true ones in here.
I was waiting for the true
Yeah.
one. Yeah.
Oh, here comes one. Cardiomyopathy is a type of heart failure.
True. Slightly qualified. True.
Okay. Once you've had a genetic test, the result never changes.
False.
Family members without symptoms don't need screening.
False.
You can't exercise competitively with cardiomyopathy.
False.
Cardiomyopathy can present at any age.
True.
Yay, we ended with a true.
There we are. Great.
So I thought the purpose of this podcast is really just to unpack some of the language really around genetics and cardiomyopathy, because I know sometimes it can be quite complex. So I thought we'd look at some of the terms that we use, and starting with you, Ruth. DNA, chromosomes, genes. What do those terms mean, and how do they relate to each other?
Big subject.
I know.
Big subject.
I know, and I'm asking you to be brief, which isn't an easy
Yeah.
thing to do, is it? But
So I guess in this context, so if we're talking about cardiomyopathy, we're talking about DNA and chromosomes, we're talking about the structures really and the substances that genes are made of. So DNA is the substance, the chemical, if you like, that genes are made from, and chromosomes are the packaging, the way that DNA is stored in the cells. So we've each got our unique DNA genetic makeup. It's a single, it's a long strand of DNA coiled up, packaged into chromosomes, which are the bit that we can see if we have a microscope. We can look and see chromosomes. And they have a very characteristic pattern. They have a characteristic number. And we have our 1 to 22, and then X and Ys that are sex determining. So chromosomes were discovered long before DNA was,
Mm-hmm.
but DNA is at the heart of any of the genetic testing that we do.
Okay. People often use an analogy of a library, don't they? Can you explain that to people, how the library analogy works?
Yeah, I think it's a nice analogy for everybody having a set of genes. So we don't know exactly how many genes there are, but probably around about 25,000. We've all got copies of those genes.
Mm-hmm.
And I guess the library is that, you've got them all there and you can pull one out and look at that particular gene. But I guess it also alludes to the fact that we've got a DNA sequence, and the sequence is the letters of the DNA code. So your entire genome has this 3 billion letters. It's actually 4 letters repeated in different ways.
It's really hard to just conceptualise that, isn't it? It's really hard.
Very hard. Long pieces of DNA we don't yet understand the function of. But, in that sense, when we're looking for changes in DNA, we're looking at changes in lettering.
Okay.
That, again, I think the analogy of the library is very good. So you might be going for one book, you might be going for one page in that book, and you're looking for a spelling mistake on that page.
I remember when we spoke before that I was saying to you that those letters just seems very random, that you've got A, T, G, C, letters, but they actually relate to something, don't they? Can you just briefly...
So they stand for those, so arginine, guanine, cytosine and thymine. So they aren't picked randomly. They're not picked randomly. Yeah, that was good.
That was really good. Well done.
Rolled off the tongue.
Yeah.
Yes. So those are the bases that DNA essentially is constructed from.
Okay. Perry, genetic and genomic, what's the difference?
It's almost philosophical sometimes, that question, isn't it? Genomic is the entire genetic story, if you like, within a cell. So it's the code, as we've been talking about, but it's the things that control the cell's ability to read that code, the influence of genes on proteins. It's the whole package. It's what genomics is from beginning to end, what genes do and how they do it and how they're regulated.
Okay.
I think genetic is a bit more specific. So gene equals protein, not necessarily that whole story.
Okay.
Is that fair?
Yes, and but I think now we this is semantics really, and genomics is everything, and genomics is where we are today. It's, as Perry says, it's not just the sequence, it's how those genes work, how they're regulated.
Okay.
And I think more and more...
So it's a better term, really.
Genomics. Yeah, I think it's a contemporary
Tells a bigger story.
term. It's a bigger
big
story, a bigger,
okay.
bigger picture.
Okay. Ruth, polygenic, monogenic, what's the difference? And also take in variant, mutation and trait.
Okay. Monogenic, the easiest. So monogenic means single, mono for single gene. So these are single gene disorders often we're talking about. There's a specific gene. You have it or you don't. If you have it, you have the disease. If you don't have it, you don't have the disease.
Okay.
Slightly oversimplified there, because even for monogenic diseases, there may be other influences.
So that's interesting, isn't it? Because actually oversimplifying is kind of, I think, probably what we need to do for this podcast, just because it is so difficult sometimes, isn't it, to pin these things down, which is why I did those questions at the beginning, because I think sometimes it's difficult to say yes or no, because there's a story behind it. So simpler the better, I think.
There is. It is an important distinction for inherited cardiac conditions and cardiomyopathies that is somewhat different from other areas of genetics.
Really?
In
How's
that
that?
I think monogenic rarely applies. We are aware that actually polygenic, multifactorial is a big part of the story with cardiomyopathy and with other inherited cardiac conditions, in a way that it perhaps isn't with certain other.
Is that a change in thinking, Perry, though? Because, if you think back to the first discovery of a gene causing hypertrophic cardiomyopathy, it was monogenic, wasn't it? It was one gene.
It was, but I haven't really thought of this point before, sort of comparison with other areas of medicine, because I think the way in which sometimes genetics are performed in the clinic, depending on the person's background, the healthcare professional's background, they may think like a polygeneticist or they may think like a monogeneticist.
Mm-hmm.
And sometimes that alters the emphasis of the information which is being transmitted. So I think in cardiovascular, we tend to think like monogenomic kind of people. And for us, the polygenic thing we bring in if we're trying to explain why there's variability in expression, why some people may still have a genetic predisposition without being able to identify a gene. But the monogenic story is very cardiovascular. I hadn't really thought of it in that way before.
I think one of the reasons for thinking differently is that we do have clear evidence in cardiomyopathies of other factors being involved.
Mm-hmm.
And that challenges thinking in that way. And I think we have less evidence for that in other diseases. It may well be the case, but we just have less evidence. But I think it's really important if we move towards personalised medicine centred around someone's personal genome. Why would it be that a single gene out of the 25,000 acts alone?
Mm-hmm.
And why would a single change in a letter of the DNA code not be influenced by other changes?
Other things, yeah.
Yeah.
Okay.
Can go down a rabbit hole with it as well, can't you? Because it's these are two extremes, but there's stuff in between as well.
Yeah.
Yeah.
And we're increasingly focusing on intermediate effects now. It's a spectrum.
Yeah.
If you were explaining, like if I came into your clinic, let's say, Perry, and I was completely, this was entirely new to me, how would you sort of go about, I mean, it might not even come into your mind to describe the difference between polygenic and monogenic, but, how would you go about describing that to a patient, the difference?
So I don't put it in those terms. It's more the consequences of the genetics. So, you've got a genetic abnormality which is predisposing you and your relatives to disease potentially, but the expression of that's going to vary. It's normal for this to vary. That's one of the questions I think we often ask. And the skipping thing that you ask, people often say, well, I've been told it's skipped. Well, I think your hesitation was appropriate because what we call the phenotype, the consequence of a genetic abnormality, what you see on an echo or an ECG, may be very different, may be extremely subtle, but it doesn't mean the gene has skipped. It just means that maybe the phenotype is so benign in some individuals that they appear unaffected.
Yeah. Yeah, and in fact, thinking about polygenic and multifactorial inheritance helps us to understand why people in families do present differently.
Yeah.
Why it might look like it skips a generation.
That term multifactorial.
You don't like that, do you?
I really don't like it. I know. It always feels to me like, I don't really know, but it's a good word, to sort of, it's multifactorial, But, what do you mean when you say that? And what are those factors that, alter your decision or your diagnosis?
Well, I think that comes back to knowing that the DNA sequence is the very most basic part of the genetic story.
Okay.
That what happens to those genes, how they work, how the disease develops from those alterations, how does it affect the cells? We know that is very complex, that these are complex pathways with numbers of different genes and indeed other factors.
Yeah.
So I guess using the word, it means if we use polygenic, we're specifically saying the effect of multiple genes. In multifactorial, we're talking potentially about what's loosely called environmental, which is not just environmental in sort of global sense, but environmental in the body.
Yes, your own internal environment.
And how that will affect the way in which genes are expressed.
Yes.
It doesn't alter the DNA sequence, but it alters the way in which those genes are expressed.
Yeah.
It's also dynamic, so it changes with aging, or the acquisition or the emergence of other comorbidities, hypertension, diabetes.
Yeah.
All of these things are going to influence how a gene expresses itself.
Okay. Variant, mutation, trait.
I think variant it's now the preferred term for a DNA sequence change.
Why is that?
I think people find the word mutation tricky for very obvious reasons.
Yeah.
It's a word that is used in genetics, not just human genetics, but across the board. I think people find that it has a kind of connotation that people are less comfortable with. But it also implies a clear understanding of that DNA sequence causing harm. And actually many variants we don't know. So this, challenging word, a variant of uncertain significance.
Your favourite?
We're holding our hands up and saying we absolutely, we're going to say we don't know.
Yeah.
But even the ones where we think we know, we may not be actually quite as sure. So keeping the word variant, it's telling you there's a difference. So what we would expect to be the sequence at that place in the gene is different. That could be your family background, could be your ethnicity, it could be potentially completely benign and doesn't actually alter the way the code is read. It doesn't mean anything. It's just a difference. But it could be that it alters everything in a really significant way, and we would call that disease-producing.
Okay.
So we tend to use the word pathogenic variant, where we are confident that the variant does something, pathogenic meaning harmful.
Yes.
And we use the word benign.
Disease-producing is probably an easier thing to understand, isn't it, than pathogenic necessarily.
We like our classical.
You like your terms, don't you?
Though, don't
No,
we?
I understand that. I understand that, but sometimes you have to unpack what they mean.
We do.
To people like me who are saying, what?
Yeah.
You
We
know?
fall back too easily into those terms because they're the terms that we learn.
You learn, it's
medicine.
natural. Yeah.
But I think we're always aware when we're talking to patients and families, try, if we can, to use terms that are more meaningful.
I agree with that, but I think people are coming better prepared now.
Yeah.
I mean, I think the internet is it frees people. They do their own research, and by the time they come to see people like you and me, they've already acquired an awful lot of knowledge. I think it's interesting, the word mutation, isn't it? Everything you said, of course, is correct in the way we should go, but mutation is a nice, simple fallback term because otherwise you're talking about pathogenic, likely pathogenic, VUS, hot VUS. It does get a bit complicated sometimes.
A hot VUS?
A hot VUS.
What's a hot VUS, Perry?
Well, because as Ruth's implying, that even things like VUSs, there's a gradation of certainty or uncertainty. There's some supporting knowledge, but not enough to push it over the line to likely pathogenic.
Okay. Wouldn't that make it cool?
Well, a cool VUS.
Yes, it would. It would.
Okay.
I think we are very lucky in the UK that we have molecular genetic laboratories that take great care to ensure that the results that they issue are robust and contemporary. And they, in collaboration with scientists in the USA, came up with a scoring system.
Okay.
Which aids in understanding whether or not variants are relevant.
Okay.
And that's where these terms have really come about.
Okay.
So likely pathogenic. So it's easier for scientists, I think, in the laboratory to classify variants. It's a bit different when you bring it to the clinic.
To the clinic and explain to patients. Yeah.
And explain to patients, yes, why likely pathogenic, not clearly pathogenic. That can be a discussion where both Perry and I work closely with scientists and are able to review these variants, and we bring the clinical information to help with the interpretation of those, and that can be where the answers lie. Actually, it's in the detailed
and this is
clinical
the detail
information.
on the question I think you asked me about whether a variant today is a variant tomorrow. It can change as more information accrues and can go either way.
Okay.
Yeah.
Okay. And trait. So I'm thinking of traits in terms of hair colour, eye colour, nose length.
Yeah, we use the word trait much less.
Really?
Now, yes. I think that is a sort of quite a sort of old genetics term.
Okay.
Trait. So I think I mean, yes, you're right, height, hair colouring. Perhaps we more apply it now to features that are universal features. So height is a universal feature. Everybody has a height.
Mm-hmm.
There's a huge range.
Yes.
Rather than the old-fashioned interpretation, which would be actually of a particular condition or disease.
Really?
Yeah.
Oh,
yeah,
okay.
Yeah. So I think it's a word that's sort of fallen
Out of
use and has been replaced by, I sometimes use the word disease, disorder, condition. Trait implies sort of normal variance.
Mm-hmm.
Normal variation.
Yeah.
And that's not really what we're looking at. We're looking at diseases. We're medical geneticists, and we're
pathogenic side
we're
of
interested
things.
in healthcare and
Perry. Mm-hmm. Inherited and familial.
I think they're the same thing. You're going to tell me they're not, aren't you?
No, I like it.
I think they're the same. I think for a patient, they're the same. Inherited means you've acquired your condition, trait, whatever we're going to call it. But the disease that you're screening this family for from a parent or from both parents, I would say that's for the individual who's inherited that, they've inherited it as a consequence of a DNA abnormality in one or other of their parents. Do you think that's reasonable?
Yes.
I think familial means that, I suppose it's more the potential for something to be transmitted. So this is a familial disease, so it's a disease which can be, it's genetic in origin and can be transmitted. Mm-hmm. I don't know whether I've just created a difference where there isn't.
I think actually the misunderstanding that occurs is between genetic and inherited.
Don't preempt my questions. Perry does that all the time.
So
don't go there, Ruth.
Is something genetic or is it inherited? Or
we'll come on to that.
Or familial. Or familial, exactly. Yeah.
Perry, whole genome sequencing.
So you have your DNA, and the DNA code is broken up into bits. So genes are particular bits of the DNA. So if we're doing genetic testing, we can look at the gene, or we can look at the bits in between, and other elements of the code that you wouldn't necessarily do in routine standard diagnostic genetic testing. So whole genome means you do the whole lot.
Right.
Other terms like whole exome means you're just looking at the bits which are coding for genes.
Okay.
And you can go down to single letters in the code if you want to. So it's the level of fidelity, if you like, of examining the genetic code, and whole genome means the whole lot.
Okay. Do we do that very often? Whole genome?
Increasingly, but...
Yeah, well, certainly in, again, UK, maybe groundbreaking, literally world-beating. We do whole genome sequencing as first-line investigation.
Do you do it
for
routinely?
For a number of rare conditions.
Right.
Not for cardiomyopathy.
Right.
Except in exceptionally rare and unusual cases.
Mm-hmm.
But the direction of travel with genetic testing is for whole genome sequencing to be the basic test, first line test. That the advantage of that is you have it done once,
Exactly.
and then that's your library, and you go and check out the bits that you want to.
How does that relate to you saying things change over time if you have it done once and that's your library?
The interpretation of a variation changes.
Okay.
Yeah. But also what Ruth is saying is you do it once, so you've built your library, but in terms of what you analyse and feed back to a patient is usually not the whole thing.
Okay.
So for diagnostic testing, you may only look at a bit of that code, but it's there on file.
Right.
So you don't have to come back and resample them. I think that's the huge advantage of whole genome.
Yeah. Ruth, penetrance and variable expression.
So I think variable of expression is easier to explain. It's the fact that, things look very different in individuals. And the important context of that is within a family.
Right.
No two individuals with the same gene necessarily have the same disease. Very confusing, I think. But that brings in all the polygenic, multifactorial, et cetera. And the fact that, we're a combination of genes from each of our parents. Our children will be a combination of genes from a different set. So there's always other genes coming in that have an effect. So that's variability of expression means the disease could be mild or more severe, could be early onset, late onset. Penetrance we tend to use it as more of a sort of on-off, yes, no. So this thing of does it really skip a generation?
Yeah.
So non-penetrance would mean that you saw no signs whatsoever of disease in somebody who did have that gene.
Okay.
The challenge with cardiomyopathy and with many other diseases is how can you possibly know that? We can't examine every cell. We certainly can't examine, technology is brilliant, but there may be cases where we can be reasonably confident that there's no evidence of disease, but I think that's not always the case.
You've also got the word, because we used to talk about incomplete penetrance, didn't we? Which I think is where it gets tricky in terms of expression. But we also have the issue as to what we define as expression of disease. So, for example, with many diseases that we have maybe put into the dilated cardiomyopathy box, when we screen relatives, we may not see enlargement of the heart size, but we might see arrhythmia, we might see an abnormal ECG, we might see extra beats. So is that a manifestation of the disease, which it probably is, but they don't necessarily fulfill the criteria for DCM, and that can get a little confusing as well, I think.
Definitely. And I think because cardiac investigations have become more sophisticated, that's good, but it also means in some ways more likely to pick up these minimal features, and then often actually the cardiologists are asking us, do you think this is a manifestation of that gene, or is this unrelated? And does this tell us anything about the gene? And vice versa? Does the gene tell us anything about what we're seeing here? Does this fit together? I mean, so much of cardiac genetics is a jigsaw puzzle. It's lots of different pieces, lots of different bits of information. Genetic testing doesn't just give you the answers from a genetic test. You need all of this additional information.
All those multi-factors. Which one of you wants to take on epigenetics?
Oh, you can do that one.
Chicken.
Lots. Big subject again, hedging a bit. Very big subject and very, very topical. I think there are a few sort of key points about epigenetics. And the main one is that DNA template is not static.
Right.
It is dynamic. And there are things that can interact with DNA. And we sometimes use the word imprinting when we talk about the way in which DNA is regulated in certain ways. There are parent of origin effects, which we call epigenetics. So whether you inherit the gene from your mother or your father, that message is imprinted differently. And this is that
even if it's the same gene?
Yep, same gene, same sequence, but whether it's paternally inherited or maternally from the mother or the father, the gene can be expressed differently. It gets different signals. So I think it's about the dynamic changes, the way in which genes are expressed, the way in which environmental changes potentially regulate DNA expression.
Okay. It's complicated, isn't it?
It is.
Even when you try and simplify it, it still gets complicated very quickly.
Yeah, very very, very complicated. And I think it was much easier in the, one gene, one protein day, few tests, few genes. Yeah, that it was easier.
Yeah.
And I think we're really into the more we know, the more we know we
don't know. Yeah.
And the practical application of that is, you said, does a variant stay the same? The variant stays the same, but the interpretation changes.
Yeah.
And the question is, how often do you need to check that out?
Yeah.
Do you say, well, that was done the last 5 years, so we're pretty okay with the interpretation? But certainly, tests from 10 years, that's a very long time ago in genetics.
Oh, that's
you
interesting.
You might be thinking, well, maybe
time to
we
be
do
retested.
need to. And we use our lab a lot to verify and reclassify.
Do you do
tests.
that, or do patients come to you and say, can you retest me now? Or do you do it routinely?
A bit of both, but I think, if we're honest, it doesn't take place most of the time. I mean, I can remember us having these conversations years ago about how do you engineer a recall process, for example.
Yeah.
So when new information becomes available, how do you then find all the people that you need to find? And so, and I don't know if there's analogies in, for example, cancer testing, but I don't think it's done very well. So, so it's often opportunistic. So someone will come back for a clinic visit. If someone remembers to look at their gene, they may go through the process of checking whether it's still pathogenic or not.
You said before, Ruth, haven't you, that from your perspective, it's a point of care thing. At that point of care, you sort of check, as the clinician, you check, when did this person last have a gene test? Is it appropriate to have another one now?
Particularly when it's going to be used for medical management. So if you're going to make a decision on the basis of that variant that affects either therapy or prenatal or other reasons for knowing about that variant, it's absolutely imperative to review. If it's not altering case management, if the family has been well worked up, most people have been tested, it may be less relevant. But we do leave it to cardiologists and geneticists to kind of initiate this. And I would be very happy if patients did as well, taking into account the fact that things change over a period of time, and that might be relevant.
This is one of the elements of counselling, isn't it?
Yeah.
I mean, I think trying to transmit that knowledge that things might change. So we think we've got it. And sometimes this is something that's been seen over and over and over again, so the likelihood of it changing is but if it's a new variant that hasn't been described before, or even a new gene which is only just been described, then things can change, sometimes quite radically.
Again, we're lucky we work within a global community that stores information about genetic variants into databases, and we're, the more testing that's done, the more information there is, and that informs the next test.
Yeah.
So, as Perry said, if this is something that's been seen frequently in lots of families with the same condition, that's quite easy. But if you have a variant in a new gene, just been identified only in a single person in the family, it may well be that you do need to review it. And as Perry said, we don't have a systematic recall system. The laboratories have been very good, but it's not complete.
This is one. I'm too old to understand AI really, but this I think is a perfect application for AI because we can't carry all this information around in our heads about the range of variants there are, whether they're pathogenic, like pathogenic. So we need the databases, which need to be iterative and constantly updated, and you need, in a way, a machine to say, when the patient comes, this variant has changed its designation or its classification. I think that's something we desperately need, I think.
Okay.
AI would be good at that. Would be able to.
Exciting times.
They do that very easily. If they knew what the variant was, they'd be able to.
Is somebody not doing that already?
Oh, I'm sure.
I'm sure they are, but people are looking. I think they're applying AI to the wrong areas, which is probably another podcast. But this is about information management.
Yeah.
in a way which you can actually empower the patients themselves.
Definitely, yeah.
Because if you've got a universal database, then why can't the patients themselves access
They
that?
can. They can.
Yeah.
The database that we use, ClinVar, is accessible to anyone.
Really?
Yeah.
Yep. Patients can check out their own variant. They
Oh,
can
wow.
see what other cases have been logged,
Okay.
where they are in the world, what the evidence was to say this is true or not.
Fascinating. Perry, in the quick fire questions, I asked you if all cardiomyopathies were genetic. If the answer to that is no, what's the cause in those that are not genetic? What causes the cardiomyopathy?
I think you asked Ruth that question.
Yeah,
actually,
I
but
had
yeah.
to answer.
Did
that.
I?
Yeah. So I was listening, you see.
Well done for listening.
So cardiomyopathy means a bad heart muscle, which is about a bad heart muscle which isn't caused by a blocked artery, heart attack, or high blood pressure. So common diseases which can also damage the heart muscle. So it's everything else. So it is possible to damage your heart muscle, for example, if you contract a viral infection which affects the heart and you have inflammation, damages the heart muscle, and you're left with a dilated heart or a scarred heart. And that would be called a cardiomyopathy, but if you like, it's an acquired form.
Yes.
I suppose you could also interpret the question as someone who has a cardiomyopathy for whom we cannot find a genetic variant. So we often, sometimes the term we're using now is gene elusive, which adds a certain air of mystery to it for some reason. But so we've done the tests, can't find anything. And that could just be an extreme part of normal variation.
It's tricky, that, isn't it? I think that's really tricky, especially when you're on the receiving end of that. It's like, because there's all this talk, as evidenced by what we're doing now, about the genetics of cardiomyopathy, that if you haven't got a gene, is it just because you haven't found the gene yet?
It may well be.
Or is it that it's not a genetic cause?
Or it could be polygenic, and it just means you've inherited a large number of variants which on their own don't have an effect. But if you've inherited a lot of them, you're now predisposed to, say, developing a thick heart should you develop high blood pressure.
Okay.
So I suppose it is a genetic mechanism in a way, but that's a much more gene-environment interaction.
Yeah.
They're going to be gene elusive if you look for the monogenic disease. It is possible to potentially say whether or not you've got lots of those common variants by doing people are creating what we call polygenic risk scores, for example. But a lot of the people we see, particularly, for example, with hypertrophic cardiomyopathy, a lot of the gene elusive ones tend to be older, male, obese, high blood pressure, maybe some sleep apnea. And so that is probably the combination of factors which will tend to drive thickening of the muscle, maybe with a genetic predisposition because of this polygenic effect. So a lot of the time when we say this is genetic or non-genetic, sometimes the non-genetic stuff has got a genetic influence to it.
Okay.
One of the most dramatic changes I think we've seen recently, for example, is in people who do have inflammation in the heart muscle, what we call myocarditis. 10 years ago, would we have started thinking about doing genetic testing in those patients? No. But we've realised actually a lot of them have monogenic disease, and myocarditis is part of their disease expression.
Yeah.
So we're trying to do more genetic testing in people with myocarditis.
When I teach the medical students, I talk to them about, pretty much everything is genetic, really, isn't it? I mean, infection is probably genetic. Why do some people develop serious infection when others don't? And I think the thing about gene elusive is it's very frustrating for families when you can't find the gene. And it's really important that they don't go away thinking there isn't a gene there. It just means we can't find it. And for conditions that we have a really high level of certainty are genetic, we still can't always find the relevant gene.
Okay.
And the reason for that is technology.
Right.
And it's getting better and better. And one of the changes that we talked a little bit about, whole genome sequencing, WGS, one of the, this is sort of, just raising the standards and levels of detection. And one of the reasons it does that is it goes over the same sequence again and again. So a bit like reading through the book. You read it once, you might miss the spelling mistake, but if you go over it 30 times, there's a greater chance. Added to which is this back catalogue of all the work that's been done before, meaning it's easier to sift through and understand, you know, is that a hit or is that not a hit?
Yeah.
And so gene elusive, you know, it drives research. That's where, you know, we would want genetic research to focus. Probably our genes we haven't yet discovered. We may know what the genes are, but we don't necessarily know all of the diseases they're implicated.
Yeah.
And we've only touched the sides of that regulation, as we call it, the gene regulation.
Okay.
So for many of these families, it may be that we'll never see this in that DNA sequence. What's happening is changes in the way in which the genes are being switched on and switched off.
Okay. So I guess leading on from that, how important is getting a genetic diagnosis, Perry? Why is that genetic diagnosis important for a patient?
So I think that operates on a number of different levels. Until relatively recently, it was, I suppose, confirmatory. So this gene matches the disease that you have, so we know that you have a genetic form of hypertrophic cardiomyopathy. I think it certainly enables greater certainty in a family and offers the possibility of screening other family members.
Yeah.
Some of whom may be carrying that genetic variant but not expressing it, as we were discussing earlier. So you're triaging those individuals within a family who need longitudinal follow-up, for example. I think it would be fair to say that until relatively recently, it didn't have a huge impact on clinical management for many of the cardiomyopathies. For some, there are lifestyle adjustments.
Yeah.
If you know that you're a variant, you can make certain life choices. So if you're carrying a variant, but you've always wanted to be an airline pilot, you need to understand that in 10 years' time, you may express the disease and then your career is over, or if you want to be a professional sportsman.
Okay.
Those kind of considerations. I think what is changing very rapidly is that we are now using the genetic information itself to make a therapeutic decision. I would say that that's probably most common in the context of dilated cardiomyopathy, well, that group of diseases. Because we now recognise that that's a very broad church of many different disorders, some of which have a particularly high risk of sudden cardiac death. So knowing that someone carries a particularly high-risk variant, you will monitor them more closely, and as they develop their phenotype, you may have a much lower threshold, for example, putting in a defibrillator. I think what we're all hoping for, and what is now starting to happen, is we're now starting to imagine targeted drug treatment, or maybe even genetic modification, so gene therapies, as we call them in very broad terms, based upon the identification of a genetic abnormality. Now that's emerging. Some of it may still be science fiction, but I think it's pointing in the direction to where actually we could actually start treating the underlying cause of the disease very directly at a genetic level.
Yet again, Perry, you've preempted my question.
Excellent.
Bit of a habit, isn't it? Bit of a habit.
They're such good questions.
Smooth. Ruth, this is what I didn't want you to go into before, but when we think of something being termed as genetic, by definition, we tend to think that means inherited, but that isn't the case, is it? Can you explain a little bit what the difference is there?
Something being genetic just means that it involves genes. And more often in this context, we're saying an individual has a change in a specific gene that is linked to their disease. That may not have been inherited from their parents, which would be inherited. Where do these changes occur? These changes occur during the formation of eggs and sperm. That entire DNA sequence, the 3,000 million letters, have to be copied faithfully. It isn't. There are lots of differences. That's why we're different, not clones. And if that's when these new mutations occur. So the idea, we all carry multiple changes in our DNA. It is different from our parents. The basis of DNA fingerprinting.
Yeah.
One of the reasons why we are unique, we can use that in DNA fingerprinting.
It's crazy, isn't it? Because you say something is genetic. Just it really feels that by definition that means I've inherited it.
Yeah.
It really does feel like a bit of an oxymoron.
Absolutely. And people's response to being told it's genetic, they'll look to their family and say, "But
who did I get it
my
from?
My parents were fine, and there's nobody in the family. It can't be genetic."
Yeah.
No, it isn't, but it is genetic here, and now that's the explanation. And for future generations, that then can be passed on and inherited into the next generation.
Yeah, that's important as well, because although you may not have inherited it from your parents, you may be the first in the family to have that genetic variant, you may now be able to transmit that to your offspring.
Yeah.
So this would be what you call a de novo mutation?
Yes.
Or a de novo
pathogenic variant.
Yes.
Yes, de novo pathogenic variant.
Yeah, okay.
Much more difficult to assess pathogenicity when it's de novo.
Yeah, I can
For
imagine.
some variants, easier for others.
Yeah. Complex.